Hello, everyone. In this chapter, we are going to address gene expression. We are going to go from DNA to protein. We are going to take a look at the different steps as we go from the genetic information contained in DNA to the production of a protein. Now, as we go through this, we are going to take a look at the relationship between proteins and amino acids. You know that amino acids are the building blocks that make up proteins. So chains of amino acids basically are called proteins. We will also look into the relationship between genes and proteins. How do we go from a language of nucleotides sequence to a language of amino acid sequence that we find in protein? We are going to take a look at all the players that are involved in that whole process. From DNA to RNA, we have a process called transcription. Then from RNA to protein, we have a process called translation, right? We're going to take a look at what are the players involved in each of the process. What are the different types of RNA molecules and what is their function? What are the transcriptional factors? Because yes, you have transcription, but what are the transcription factors that control the production of the transcript, right? We are going to dive into the steps of transcription. Then we are going to dive into how the genetic code was discovered and what it means in terms of going from DNA sequence to amino acid sequence to eventually proteins. Then we are going to take a look at the structural levels of proteins. And finally, how do proteins after production get folded so that they can adopt their ultimate three-dimensional shape that is compatible for their function? And what happens when these proteins are not taking their proper folding? All right. So all the proteins that we ingest, say for instance you are eating a T-bone steak or a sirloin. A sirloin or a T-bone steak, it's nothing but muscles. These muscles are a bunch of proteins, not only proteins, but we are focusing on proteins, right? So these are a bunch of proteins. These proteins in your digestive tract are going to be broken down into amino acids. The amino acids are the nutrients that your body is going to absorb and use for the production of your own proteins. So all the protein that you ingest, there is no way these proteins are going to go from your stomach or your small intestine to directly your system. This is not going to happen, right? These proteins, when they come in your system, in the stomach, they get broken down by some by some enzymes called peptidases, right? In a small intestine also, the proteases. The proteases are going to break down proteins into small chunks of sequences of amino acids. These small chunks are called peptides. These peptides are going to be further broken down by peptidases. These peptidases are the enzymes that are going to cause a release of amino acids, which are the building blocks of proteins. And again, these amino acids are the one that are going to be used to synthesize your own proteins. Now, when we consider the different amino acids, because they occur in different type, this amino acid occur in 20 different kind. This 20 different kind of amino acids are found in our system. However, 9 of the 20 different amino acids that occur in our system are considered essential. Now, what we consider essential are these amino acids that we have to obtain by the food we eat. Our body may not know how to synthesize these amino acids. We have to obtain these amino acids from the food we eat. That's why we call them essential amino acids. Now, with that being said, what is characteristic of amino acids? Amino acids are characteristic by the fact that they all, without exception, have an amino group. Right? That amino group, it's basically nitrogen, hydrogen, and hydrogen. All right? To that amino group, you are going to have also in an amino acid, an acid group. That acid group, it's COOH. Then you're going to have a side chain. That side chain is called the R group. That R group, it's what varies from one amino acid to another amino acid for the 20 different amino acids that exist in our system. All right? So that side chain is going to vary from one amino acid to another. Now, 2 amino acids are going to come together through a covalent bond to form a dipeptide. From a dipeptide, we are going to have another amino acid that is going to come and attach. Now we have a tripeptide. From a tripeptide, we have another amino acid that comes and attach. Now we have a tetrapeptide. Whenever we have a long chains of amino acid attached to each other, rather than counting each of them, we are going to say that we have a polypeptide, right? So a protein, it's nothing but a polypeptide, which is chains of amino acids. So this is an example I'm drawing here. Okay. Just trying to show you something that is a chain of amino acids, right? This would be a polypeptide, even though we can count the amino acids here. And again, here, this is a peptide bond, okay? Peptide bond. All right? So these proteins, after they form a long chain of polypeptide, they are going to fold because of the charges that exist in some amino acids. Because some amino acids are polar, the chain is going to fold into a 3D shape. That 3D shape determines the conformation of the protein, and that conformation is essential for the function of that protein. Now we can ask then, what are the different functions of proteins? Proteins play so many different roles in the cell or in the body. Some proteins are going to be specialized in allowing movement. When you think, for instance, of you running or walking, that movement, proteins are the one that allow that. Some proteins are regulatory proteins. In other words, they are going to control what is being expressed when. If you consider, for instance, a 5 years old boy, a 5 years old boy produces testosterone, right? He's a boy, he produces testosterone. However, a 5 years old boy does not have facial hair, right? You wouldn't typically see a 5 years old boy with facial hair. What I mean by facial hair, it's a beard, right, or a mustache. A 5 years old boy would not have a mustache or a beard. Why? Because the amount of testosterone that is produced is just too small. However, when the child reaches puberty and becomes a young adult, the amount of testosterone increase in its production, right? And that increase in the production, it's basically a number of regulators that were suppressing the expression of the gene responsible for testosterone production to actually allowing these genes to be expressed, right? So you have some proteins that control when some genes are being expressed, when some are not being expressed. Some proteins are enzymes. They catalyze metabolic reactions. Others are structural proteins. These structural proteins are, for instance, if you consider your nails, right? Your hair, to name only these two, these are structural parts of your body, right? They describe a frame in your body. Then you have some that are going to play the role of transporters, right? Now, the transporters, if we think, for instance, of oxygen that we breathe in, hemoglobin is going to catch that oxygen and carry it through the body, right? Now, hemoglobin is not the only protein that does transport. You have many other proteins that do transport. Then you have some proteins that are going to be involved in protecting you against infectious particles, right? You have some protein that plays a role in immunity. These proteins that are also found in the plasma are called globulins. Now, the globulins that are part of your immunity are going to be called immunoglobulins. Okay? And then you have some proteins that are going to be involved in blood clotting. Now, all the proteins that exist in your body represent a number of segments of DNA that translate into protein. All the segments of DNA that translate into protein altogether, we call that exomes. So these are all the encoding part of your genome that translate into proteins, right? We have about 20,000 plus proteins in our body. Now, when you consider the proteins in our body, remember we said we have 20 different amino acids, right? So when we compare the proteins, the amino acids that occur in these proteins are very similar. The only difference is the sequence of these amino acids. In which sequence are they attached one to another to another? Eventually, that sequence is going to translate in terms of the conformation or the 3D shape, right? Which is going to cause one protein to be an enzyme, another protein to be a transporter, another protein to be a regulator, and so on and so forth. All right. So, what do we call gene expression? Gene expression, it's basically the process by which we go from DNA to protein. The cell is going to use two processes in order to produce proteins from the information that is contained in DNA. The first process is transcription. Transcription, it's basically converting the language in DNA into another sublanguage that is found in RNA molecules. Now, DNA and RNA are both nucleic acid molecules, right? However, we know that DNA contains as nitrogenous bases adenine, guanine, cytosine, and thymine as nitrogenous bases. As sugar, a 5-carbon sugar, deoxyribose. However, RNA is going to have as nitrogenous bases adenine, guanine, cytosine, and uracil. In terms of 5-carbon sugar, RNA is going to have ribose. So you see that even though the building blocks in DNA and RNA, we call them nucleotides, however, these nucleotides are different, right? They are different based on the type of sugar they have, right? One carries a ribose, the other one carries a deoxyribose. This difference is significant enough to make RNA and DNA two distinct kind of proteins, even though they still fall in the category of nucleic acids, right? So transcription is converting the language of DNA into the language of RNA through a process that we call transcription. And then the information now that is transcribed is going to be translated. That translation is going to allow to convert the information in RNA into another type of language that is the language of amino acids. So while reading the information in RNA, we're going to be able to find equivalent in amino acid so that you are going to go from a nucleotide sequence to an amino acid sequence, right? And eventually, the outcome of that is going to be a polypeptide that is going to be folded in 3D and obtain its final conformation. And when it obtains its final conformation, now the protein can be active. Now, I want you to remember that the 3D shape is the single most important aspect in a protein to be functional. If a protein is not properly folded in its 3D shape, that protein is not going to be functional. Let me repeat that. If a protein is not properly folded in its three-dimensional shape, that protein will not be functional, right? So this is the reason why integrity of proteins in terms of shape can affect its function. Now, there are some environmental conditions that can affect the integrity in terms of shape and also affect the integrity in terms of function. We call this denaturation. Denaturation. Loss of shape equals loss of function, right? What are some conditions that can cause denaturation? We're going to have a tremendous shift in the temperature or a tremendous shift in the pH. But let me close that parenthesis and let's come back to gene expression. So, in gene expression, we have what we call the central dogma. What is the central dogma? The central dogma says that when you consider a cell, DNA being the genetic information, that genetic information in DNA is going to be first and foremost converted from DNA to RNA, right? From DNA to RNA, and from RNA to protein. This is one-way flow of information. So we have a directional flow of genetic information from DNA to RNA to protein. That's the central dogma. Another dimension of the central dogma is that for one gene, you have one protein. That's another form of the central dogma, but I'm going to close that parenthesis for now. Now let's take a look at RNA. Remember, in the previous chapter, we looked at DNA. We spent a tremendous amount of time on DNA, so we're not going to come back to that for now. Now let's take a look at RNA. What is RNA? RNA is ribonucleic acid, right? Ribonucleic acid. This is a single-stranded molecule, right? As opposition to DNA that is a double-stranded molecule, right? RNA, however, when it comes to gene expression, it represents actually the bridge between the gene and the protein. You have no proteins without DNA, without RNA. Okay? No protein without RNA. So, in other words, the basis of an RNA are going to be used to produce proteins. And this information in RNA is complementary to one of the strands in the double helix DNA molecule. Typically, an RNA sequence is going to be complementary to what we call the template. Now, the template is what is being read, right? What is being used to synthesize RNA. However, what is going to be interesting is that the template, it's not the part that carries the information. The part that carries the information is the opposite strand, what we call the coding strand. This is very important to understand because whenever the cell wants to express the gene, the cell has to know exactly which strand of the DNA carries the gene, right? So rather than reading the strand of DNA that carries the gene, we are going to read the complementary strand of the one that carries the gene so that you are going to produce a sequence that is exactly the same as the one that carries the gene. All right? It may sound confusing, but that's the way it works. If something is confusing, please let me know. You have one enzyme that is responsible for reading the template strand and converting that template strand into RNA. That enzyme is called RNA polymerase. Okay? RNA polymerase is the enzyme that produces RNA molecules. Now, going back to the central dogma, we have DNA. From DNA, we produce RNA. In the process of transcription. And then from RNA, we are going to produce proteins through the process of translation. But for now, we are going to focus on transcription. In transcription, the two strands of DNA here are going to be separated. And now we are going to ask which of the two strands is carrying the information for a protein to exist. So we have the DNA coding strand. That is the strand that carries the information for a protein to exist. However, the complementary strand is the one that is going to be used as template strand. From there, the RNA polymerase is going to actually copy the complementary strand of this, right? And what you are going to see is that by producing an RNA that is complementary to the template strand, you are producing exactly the same information as the one on top. For instance, if we look at the sequence here, T, G, T, C, A, T, T, C, G, G. Now, remember that T, it's for thymine in DNA, but we need to synthesize an RNA molecule. And we know that in an RNA molecule, in place of T, we have U. With that being said, for that C here, we're going to have U, we're going to have G, we're going to have another U, we're going to have C, A, U, U, C, G, G. Let's take a look. We have G, G, C, U, U, A, C, U, G, U. And notice that each of this is complementary to the nucleotide here. G complementary to C, G complementary to C, C complementary to G, and so on and so forth. All right? And the RNA polymerase is the one that allows to convert, to read the DNA template strand into this sequence of RNA. All right? And now we can ask, what are the different kinds of RNA molecules that exist, right? So, before we dive into that, let's first compare DNA and RNA. DNA, usually a double-stranded molecule. RNA, single-stranded molecule. In DNA, among the nitrogenous bases, we have adenine, guanine, cytosine, and thymine, right? While in RNA, we have adenine, guanine, cytosine, and uracil in place of thymine. I want you to remember that. Then, In terms of the 5-carbon sugar, for DNA we have the oxyribose, while for RNA we have ribose. And for DNA is going to maintain the protein encoding information, while RNA carries protein encoding information and controls how the information is used. All right? DNA cannot function as an enzyme, but interestingly, RNA can function as an enzyme. Actually, one of the enzyme that is RNA is called ribozyme. There we go. And then DNA persists, can be passed down from parents to offsprings. That is not the case for RNA. Okay? RNA is short-lived. Okay? RNA is produced, then it's destroyed. But DNA is always there. All right. So DNA stores the information that is going to be converted to RNA and protein encoding information, right? While RNA only carries protein encoding information, that's it, right? Very important to remember, okay? Double-stranded molecule here, single-stranded molecule here, the oxyribose, ribose, right? The nitrogenous base, thymine, cytosine, adenine, guanine for DNA, uracil, cytosine, adenine, guanine for RNA. Please remember that. All right, question, what are the different types of RNAs? There are 3 kinds of RNAs. You're going to have the messenger RNA or mRNA. What is a messenger RNA? Just like its name indicates, that RNA carries a message. That message is basically the information for a protein to exist. That's what you need to remember. Then, the next type of RNA we are going to cover is the ribosomal RNA or rRNA. Ribosomal RNA, it's just the name indicates, is the RNA that is found as a structural part of the ribosome. And what is a ribosome? A ribosome, it's a bunch of RNA and proteins, okay? Proteins and RNA all together, they are going to form ribosome, all right? And finally, we have what we call the transfer RNA or tRNA. The transfer RNA, it's the RNA that allows the system to read the information in the messenger RNA and convert that information in the messenger RNA into information in amino acid. So a transfer RNA is characteristic by the fact that you have, let me just draw something like this. Let me up here. Okay, so you have one end here where we have an amino acid carried by the tail. And then one end here where we have 3 nucleotides that recognize a very specific sequence. Okay, so 3 nucleotides. Okay, and these 3 nucleotides are going to recognize some nucleotides on the messenger RNA. All right, we're going to take a look at some more details a little later. So other classes of RNA are going to basically control which gene is being expressed when and in which circumstance. But let me close that parenthesis. So remember, three types of RNAs: messenger RNA that carries the information responsible for a protein to exist; ribosomal RNA, the RNA that is found as structural part of the ribosomes. These RNAs are also the RNAs that carry the ribozyme enzymatic activity, right? And you have the transfer RNA, which is an RNA that carries an amino acid and that has an equivalent nucleotide sequence that matches a messenger RNA information. With that said, let's move on. Now, the different types of RNA, we're going to have messenger RNA that exist in different sizes, and these sizes vary between 500 nucleotides and 4,500 plus nucleotides. This is going to contain the information for amino acid sequence to develop. The ribosomal RNA or rRNA, they exist in size that varies between about 100 nucleotides to around 3,000 nucleotides. This ribosomal RNA associated with proteins and the formation of ribosomes, right? They also catalyze some reactions. These are ribozymes, right? That are going to catalyze the reaction of the protein synthesis. I want you to remember that. And then you have transfer RNA, which is much, much smaller. The size varies between 75 nucleotide and 80 nucleotide. They are going to transport very specific amino acids, right? So that these amino acids can actually connect to each other and form a polypeptide that eventually we're going to call protein at some point. All right. So the messenger RNA carries the information that is very specific for a protein to exist. We have that messenger RNA is going to have some coding part that we call codons. These codons are nothing but sequences of 3 nucleotides. For instance, A, G, C. AGC has an equivalent in amino acid. Another example, C, U, A has also an equivalent in amino acid. But we are going to cover that when we look at what we call the genetic code, right? Then, what else do we need to mention? From one cell to another, the messenger RNAs are going to be different. That is also the reason why you're going to have different sets of proteins. And for that reason, you are going to have some cells that are liver cells, other cells are muscle cells, other cells are nerve cells, and so on and so forth. So the information that is in the transcript is the one that is going to be used to produce proteins. The ribosomal RNA, I did tell you about ribozymes, right? Remember that? Okay? They are going to be structural part of the ribosomes. And ribosomes, they occur in 2 subunits. Now, because we are in human genetics, I'm going to address the ribosome you find in human cells, right? So these ribosomes occur in two units. You have a large subunit and a small subunit. The large subunit is called 60S subunit, while the small subunit is called 40S subunit. However, the whole ribosome together, it's an 80S ribosome. Now, 80, it's not a matter of trying to add 60 and 40, no. Okay? It's not a matter of adding them. The eukaryotic ribosomes are called 80S ribosomes, while prokaryotic ribosomes are called 70S ribosomes. But let me close that parenthesis. Now, what is the difference between the large subunit and the small subunit? The large subunit is different from the small subunit for the fact that the large subunit is much larger. And why is it larger? It is larger because it has a bunch of RNA, 3 molecules of RNA in there, right? And this RNA have a lot of nucleotides, right? 5,347. That's not neglectable. That's large, right? And along with these 3 molecules of RNA, you, sorry, you are going to have 47 proteins that are going to be complexed with this molecule of ribosomal RNA. And then for the small subunit, you're going to have a single RNA molecule that is going to be as long as 1,869 bases mingled with 32 different proteins. So this is the ribosome, and this is the organelle that is responsible for protein synthesis. And finally, the tRNA. The tRNA is going to be represented in two forms: a 2D shape that has the shape of a clover leaf, and a 3D shape that is kind of like an L shape. That's why earlier I did represent something like this. All right? Now, what is characteristic of the tRNA? It's that you have one end right here where the amino acid is attached, and another end right here where you are going to have what we call an anticodon. Now, the messenger RNA carries the information. The information in the messenger RNA is carried by what we call codon. A codon, it's a sequence of 3 nucleotides. For instance, A, G, C, or U, C, A, right? So this would be a codon. The question we are going to ask, what is the anticodon? The anticodon is going to be, if you say anti, so this is going to be U, right? This will be C, and this will be G. So you are going to imagine the transfer RNA as having UCG for an anticodon and have the amino acid right here for this AGC. All right? And this is basically how the system is going to read the information in the messenger RNA. All right? I'm going to explain with more details a little later. And I know that all of this is kind of complex, but please bear with me. All right. So you have a leaf representation right here and an L-shaped representation with the extremity 3' that is the extremity where the amino acid is going to attach. Okay? The amino acid attaches at the extremity 3', while there is an area right here that is what we call the anticodon. That anticodon is what is going to bind on the messenger RNA, right? So remember that the two codons have to be complementary, right? All right, with that said, you have a visualization, right, of what this system is going to be. All right, let me change the color here. Okay, so a tRNA, say you have a messenger RNA with the following sequence, AUG. That AUG is going to have an anticodon that is UAC. Now, for the anticodon UAC, the transfer RNA is going to carry the amino acid methionine. That methionine is the equivalent of this AUG sequence. Likewise, the equivalent amino acid for GGA in the messenger RNA is going to be glycine right here. All right? Now, that glycine is carried by a transfer RNA with an anticodon that is complementary to this sequence, right? And that anticodon will be CCU. Hopefully you understand the concept, right? Now, finally, for a messenger RNA that is UGU as a sequence, the equivalent amino acid is going to be cysteine, right? With the anticodon ACA. So now you know how we go from an RNA sequence to an amino acid sequence, right? Eventually, let's assume that this 3 molecules of RNA were all connected like this, linear. So then what we would see is that to this methionine, you're going to have a peptide bond that develops here. And then from this to this glycine, you're going to have another peptide bond that develops here. So when all of this is unattached from the transfer RNA, what you are going to see, you're going to see met, glycine, and cysteine. That's what you would see. All right? All right. So now, let's address the transcription factors. Before we address the transcription factors, let me first of all define what a gene is. Now, when you consider a piece of DNA, so say this is a chromosome, right? And you have a particular gene, and your chromosome, we're going to say we read it in this direction, all right? Now, we are only interested in the coding strand, right? You know that DNA, it's a double-stranded molecule, and it has its anti-parallel, right? One strand running this direction, and the other one running this direction. So say this is the strand that carries our gene of interest. Okay. So I'm going to say this right here is my gene of interest. But for the sake of clarity, I'm just going to say that, okay, well, this is my piece of DNA, right? Double-stranded molecule. Okay, so this piece of information right here carries the information for a protein to exist. Say it's the protein for an enzyme that we are going to call lactose. So the sequence here is going to cause a messenger RNA for lactose to exist. But here is the story. For lactose to exist, you need a number of components that I'm going to draw here that are ahead of the information that is needed for lactose to exist. That sequence that is ahead of this sequence in green is called a promoter. A promoter. A promoter actually sets the where is the gene starting, and then we have down here a terminator that sets the limit of where the gene stops. So we have the beginning of the gene and the end of the gene. In between, you have the sequence that causes a protein to exist. Without a promoter, forget about gene expression. Okay? Let me repeat that. Without a promoter, forget about gene expression. Without a terminator, forget about stopping transcription. But let me close that parenthesis. What I want you to understand and remember is that for genes to be expressed, you need a promoter. And the promoter, it's where the transcriptional factors are going to act. Let me repeat that. The promoter region, it's where the transcriptional factors are going to act, whether it is in terms of stimulating gene expression or shutting down gene expression. All right? The transcriptional factors are any factor that controls gene expression. And these transcriptional factors are going to respond to a number of signals, right? And these signals are going to be environmental. For instance, there is a bunch of sugar in the blood. The body has to internalize that sugar. It has to internalize that sugar. Now, what are the organs that are going to internalize sugar? We have the liver, we have the muscles. Now, question, it's what needs to happen for the sugar to leave the blood to enter the muscle cells or the liver cells? You need transporters. And the question is, what does it take for these transporters to be on the surface of the cell so that they can carry the sugar in? Well, these transporters, they are proteins, they have to be produced, right? They are going to be produced after gene expression, right? So the transcriptional factors have to stimulate the production of these transporters of sugar so that that sugar can be internalized in liver cells or in muscle cells. All right? So the transcriptional factors play a very important role in initiating the transcription of genes, right? They're going to respond to signals, right? Different kind of signals, but all of that, it's part of cell communication, and I'm not going to bug you into with this. Now, the last thing I'm going to mention here, it's that if by chance, let me take a different color. This one, black. If by bad luck, you have a modification that happens here in the promoter, that mutation can affect gene expression. It can be in terms of shutting down gene expression or overstimulating gene expression. But in any case, the mutations in a transcription factor can affect tremendously things, right? Okay? So that's what you need to remember. All right. Okay. So now, what are the steps of transcriptions? There are 3 steps to transcription. The first step, it's initiation. Then you're going to have elongation. So this is when the RNA polymerase is actively copying the RNA, right? And then termination, it's when there is a stop to the transcription. Now, let me take you back here. Remember, I told you that a gene, it's a promoter, a sequence that encodes for the messenger RNA, and a terminator. Now, what I did not tell you is that the sequence that causes a messenger RNA to exist, and that is responsible for the production of a protein, it's called, now, hang on, open reading frame. Open reading frame. The open reading frame, it's this information right here that is responsible for a protein to exist. Everything else is accessory to a gene. But let me close that parenthesis. So, initiation occurs when a transcriptional factor or many transcriptional factors are going to bring or foster the attachment to RNA polymerase on the promoter. Remember that. Let me repeat that. Initiation on transcription is going to happen when transcriptional factors attach to RNA polymerase and pull RNA polymerase to the promoter area. This is when RNA polymerase will start doing its job. Okay? Without the formation of the complex RNA polymerase transcriptional factors, there is no transcription. All right? And from there, the RNA polymerase will start doing its job, which is actually to read the complementary sequence and produce a messenger RNA. All right? Now, next step, you're going to have elongation. That elongation is done by RNA polymerase, okay? But that elongation also implies that DNA is being unwinded, right? You have to unwind DNA to make the template available for transcription, right? You are also going to need bunch of free nucleotides, right? And this nucleotides are actually bunch of nucleotides with ribose, right? And basically, the RNA polymerase is going to read the template so that you're going to have, at the end of the day, a messenger RNA. The terminator sequence is going to be there at the end of the gene to tell the machinery that this is where you stop the transcription. And at that point, the messenger RNA is released. Now, in terms of initiation, we have a promoter here, all right? And here we have the open reading frame that starts here. Now, that promoter area is going to have a number of signaling sequences. These signaling sequences are the ones that are recognized by transcriptional factors and RNA polymerase. All right? And one of this signaling sequence, it's the Tata box. The Tata box, TATA box. That TATA box is characteristic of promoters. Okay? So the transcriptional factor actually is going to bind on the Tata box, all right? And then you have some more transcriptional factors that are going to come and bind on the Tata box binding protein, which itself, the Tata binding protein, it's a transcriptional factor, okay? So another transcriptional factor is going to come and bind here. And as now you have this complex, you're going to have RNA polymerase that is going to come so that transcription can start. Okay? Without that complex, forget about initiation of transcription. Now, as the binding protein attaches to the Tata box and the transcriptional comes and the transcriptional factor pulls the RNA polymerase, now you have the production of an RNA. All right? That RNA is going to be a single-stranded molecule. So the RNA polymerase will keep moving along the sequence, along the template, so that a transcript can be produced, and a transcript is going to be produced in length. And at the end of the day, when you reaches a termination, this is when the RNA polymerase is going to leave the system and the RNA is going to be released. Now, in eukaryotic cells, the story is more complicated than just producing an RNA. All right? We are going to take a look at some details here. Before we take a look at these details, let me tell you that, whenever a cell needs to produce a protein through gene expression, it hardly produces a protein from one single transcript. Most of the time, you are going to have multiple transcripts, and these multiple transcripts are also going to be translated so that in a relatively short period of time, the cell can meet its need in proteins. Remember that you have structural proteins and functional proteins. If, for instance, the functional protein is an enzyme, right? You don't need one molecule of enzyme. You need multiple, right? How long is it going to take to produce those multiple enzymes? You need multiple transcripts, right? This is how the cell is going to gain time, save time. Now, in eukaryotic cells, the messenger RNA is hardly used as is. It has to be processed after being produced. So that processing tells you that the messenger RNA that is going to be converted into protein, it's not actually the initial messenger RNA. So you have what we call a pre-messenger RNA. That pre-messenger RNA is going to have to go through some processing. That processing involves the addition of a cap at one end. So say this is your messenger RNA. This is 5 prime and this is 3 prime end, right? So you're going to have a cap that is going to be added here and another cap that is going to be added here. So these caps are there to protect the information right here, okay? So that information is not destroyed, okay? And this happens before the messenger RNA leaves the nucleus to go in the cytosol, right? Now, for this to happen, you are going to need a recognition protein that attaches to the 5 prime end, okay? So the methylated cap attaches to the 5 prime end, but the kind of cap you find at the 3 prime end, it's actually a tail of multiple A's, poly-A tail. So that poly-A tail is going to be A, and so on and so forth. So even if some of them get eaten, you still have some that are going to protect the information inside here. All right? So understand that the addition of a cap and the addition of a poly-A tail are essential for protecting the open reading frame, the messenger RNA. All right? So now, in the nucleus, you have more processing than that is going to be done. That more processing, it's actually in terms of splicing. What is splicing? Splicing, it's basically cutting some information out of the pre-messenger RNA. So some information is going to be discarded intentionally, some is going to be kept, right? Any non-coding part in the pre-messenger RNA is called intron, while any coding part is called exons. So the exons are the segments of the messenger RNA that are going to be ultimately converted into protein, while the introns are non-coding parts, they are going to be removed, right? So at the end of the day, you have a mature messenger RNA that is going to be made up of only a cap, a poly-A tail, and bunch of exons. So say exon 1, exon 2, exon 3, exon 4, and exon 5. Everything that was here as intron, say you had in this area a little fragment like this, in this area a fragment like this, all of this is all gone. Okay? All of this is all gone. But you are going to understand when you see the picture in a minute. Now, because of the processing of DNA, of the messenger RNA splicing, this actually causes what we called at one time the central dogma, which is that one gene equals one protein. We are finding that, well, one gene is not always equal one protein, because the formation of the mature RNA can vary based on the type of signal the cell is receiving. So one messenger RNA mature can be exon 1, 2, 3. So 1, 2, 3. The other one can be 1, 2, and 4. That is not present here, right? So now you are going to have two proteins that are similar in here, but different right here. Yes, a lot of our hormones are actually produced like that, from one single gene, but multiple forms of messenger RNA that are slightly different, so they produce, at the end of the day, different proteins. These are isoforms of messenger RNA. And these isoforms are produced by way of alternative splicing. Okay? So the same messenger RNA is going to give different types of proteins down. It's amazing how the body works. Really amazing. All right. So now we are going back in the cell. We are in the eukaryotic cell here. We are in the nucleus. So here is the DNA, right? We have our template. We've actually a sequence with 3 exons, exon A, B, and C. Between exon A, B, and C, you are going to have intron 1 and intron 2. The pre-messenger is going to have everything, all right? Exon A, intron 1, exon B, intron 2, exon C. Then you have splicing. Well, actually, before the splicing, you first of all have a cap at the 5' end, right? Which is a methylation, and a polytail at the 3' end. And then you have splicing. That splicing is going to cause us to have a mature messenger RNA with exons A, B, and C connected together, right? But in alternate splicing, we can very well have another messenger RNA that only has exon A and C, but not exon B. Another messenger RNA that is going to have exon B and C, but not A. Another that is going to have exon A and B, but not C. It's amazing what the body can do, right? So now the mature RNA is going to leave the nucleus via nuclear pores, and in the cytosol, the ribosomes are now going to do translation. And this is when we are going to stop this video for now. Thank you for your attention.